Assalamu alaikum. I am Dr. Mohammad Ehsanul, Associate Professor of Biochemistry at the Akij Medical College, Khulna. Today our discussion topic is Introduction of Digestion and Absorption. So what is digestion? Digestion is a physico-chemical process by which complex food materials is broken down into simple absorbable unit. The dietary substance like carbohydrate, protein, fat we take are in complex form. They are not suitable for the absorption across the enterocytes. So they need to be converted into simple absorbable form. So after the digestion, there is absorption. What is absorption? Absorption is a process by which the end product of digestion, water, electrolytes, minerals and vitamins pass through intestinal epithelium to enter in the limbs or bloods. So absorption and digestion works hand in hand. So the digestion process requires some digestive juices, and these are namely saliva, gastric juice, pancreatic juice, bile, succus and uric acid. And saliva comes from salivary gland of mouth, gastric juice comes from gastric gland of stomach, pancreatic juice comes from pancreas, bile is secreted by the liver, and succus and uric acid is obtained from small intestine. And all these have different type of pH. Saliva has the pH of 6.7 to 7, gastric juice has the pH of 1.5 to 2, pancreatic juice has the pH of 8 to 8.3, and bile and succus and uric acid got almost neutral pH. So why these enzymes have different pH? Because all these digestive juices have different enzymes, and different enzymes have different optimal pH in which they work best. So let's talk about the function and composition of this digestive juice in short. First of all, we got saliva. The composition of saliva is mucus and salivary alpha amylase and lingual lipase. Salivary alpha amylase is very important enzymes, and you can break down the 1,4 alpha glycosidic bonds, but it cannot digest the 1,6 alpha glycosidic bonds. So there is a partial digestion takes place inside the mouth. As well, the lingual lipase can also digest the triacylglycerol to some extent. But we don't keep our food substance inside our mouth for a very long time. So digestion process is not very substantial inside our mouth. So what are the other functions of saliva apart from the digestion? It helps in speech, in deglutition. It also helps to gain the taste of the food. It also has some protective functions like killing some bacteria by thiocyanate, etc. And after that, we have gastric juice. The composition of gastric juice is hydrochloric acid, mucus, pepsin, gastric lipase, and intrinsic factor of Cassel. And we know there are 4 types of gastric secretory epithelial cells in the stomach. The mucus cell secretes mucus, parietal cell secretes hydrochloric acid and intrinsic factor, and chip cell secretes pepsin, and G cell secretes local hormone gastric. So sometimes examiners will ask you in the professional question how hydrochloric acid is produced inside the parietal cell. So inside the parietal cell, the carbon dioxide will attach with the water and produce carbonic acid, and this carbonic acid will be dissociated again into hydrogen and bicarbonate. The hydrogen will go into the gastric lumen, and bicarbonate will come back to the blood in exchange of chloride, and this chloride will ultimately go into the gastric lumen through facilitated diffusion and bind with the hydrogen and produce hydrochloric acid. So whenever there is a production of hydrochloric acid, there will be the increase of alkalinity in the blood. It is called alkaline tide or postprandial alkaline tide. Whenever we take a huge amount of food substance, there will be increase in alkalinity of the blood. This is called postprandial alkaline tide. So what are the other functions of the hydrochloric acids? Hydrochloric acids helps to activate the pepsin. It also maintains the optimum pH for the activation of the pepsin. It denatures the protein. It also converts the iron into the ferrous form. And for the iron to transfer across the enterocyte, you need to be converted into ferrous form. And lastly, it also sterilizes the upper GRT, and it can kill some pathogens like bacteria and viruses and keep the our body protected from these pathogens. So if the parietal cell fails to deliver the hydrochloric acid and intrinsic factor, what will happen? The iron deficiency anemia and megaloblastic anemia can be caused by this failure. So next is bile. Bile is a digestive juice which is secreted from the liver. So bile doesn't have any digestive enzyme, even though it is called a digestive juice, because it helps to emulsify fat, and it also helps to form micelles and helps the end product of digestion of lipid across the enterocytes. The composition of bile is bile salt like sodium taurocholate, sodium glycocholate, and bile pigments like bilirubin and biliverdin, and also cholesterol ester and phospholipids. So bile is very important digestive juice in terms of digestion of lipid. And in examination, examiner might ask you about the pathway of bile. The bile is secreted from the liver, and it is collected by left and right hepatic duct, and through common bile duct it goes into the gallbladder through 60 duct, and it is concentrated there, and whenever it is required, it releases the bile into the second part of duodenum with the influence of cholecystokinin. And next we have pancreatic juices. Pancreatic juice have different type of enzymes, and very important enzyme for the digestion. It got lipolytic enzyme like pancreatic lipase, colipase, cholesterol esterase, and phospholipase A2. It got amylolytic enzyme like pancreatic amylase. It got proteolytic enzyme like trypsin, chymotrypsin, carboxypolypeptidase, elastase, collagenase. And lastly, it also got nuclease, ribonuclease, and deoxyribonuclease. And last but not the least, the Succus entericus, the juice from the intestine. It also got amylolytic enzymes like maltase, sucrase, lactase, especially these are disaccharides. It also got alpha-limit dextrinase, which can break down the starch. The proteolytic enzyme also is there, like aminopeptides, dipeptides, tripeptides. And lastly, the enterokinase is very important to activate the trypsin, and trypsin can activate the other peptides. So these are all the digestive juices which is available in our digestive tract, and they are controlled, the release is controlled by nervous factor and hormonal factor. So we briefly talk about the hormonal factors here. The hormonal factors of this digestive juice are gastrin, cholecystokinin, secretin, somatostatin, gastric inhibitory polypeptide, vasoactive intestinal peptide, and these are called local hormones of GIT. Gastrin stimulates gastric acid secretion, whereas somatostatin and gastric inhibitory polypeptide inhibits gastric acid secretion. The cholecystokinin stimulates the enzyme-rich secretion of pancreas, whereas the secretin stimulates the bicarbonate-rich secretion of pancreas. And cholecystokinin, secretin, and vasoactive peptides all decrease the gastric acid secretion. So these are in short all about the digestive juices and their controls, and hope you have got the idea about the digestive juices, our digestive systems. In our next class we will discuss about the digestion process of carbohydrate, protein, and fat, and further biotransformation in the cell. So thank you everyone. Thank you for the patience hearing. Assalamu alaikum.